This video explains some basic concepts in the field of genetic engineering such as genes, cells, and DNA and related technology. As most of us know, the cell is the basic unit of life. In complex or higher life forms, within the cell is the nucleus, which contains the genome or the genetic material. The genome is usually split between chromosomes. For instance, in humans, it is split between 23 pairs of chromosomes. Chromosomes contain the tightly packaged DNA, the basic unit of genetic code. DNA segments that contain instructions for making proteins are called genes. In bacteria, which is much simpler, the DNA is contained in a single circular form. Breaking down DNA helix. DNA has a double-stranded structure wherein the two strands are intertwined to form a helical structure. Now let us understand the structure of DNA in a more simplified form with simpler representation in an unwinded form. Each strand of the DNA is a polynucleotide, that is, many nucleotide units held together by covalent bonds. A nucleotide has three components: a 5-ring carbon, a phosphate group, and a nitrogen-containing base. The nitrogen-containing base may be adenine, guanine, thymine, and cytosine. A DNA strand is therefore a string of ATCGs in various combinations. The two DNA strands are held together by hydrogen bonds between N bases. Each base forms H bond with specific complementary base, that is, adenine with thymine and vice versa, and guanine with cytosine and vice versa, and each unit is called a base pair. A DNA sequence is therefore a chemical with several base pairs arranged in two parallel strands, each strand being a series of ATCGs in various combinations, and these combinations in turn become codes for different types of proteins. DNA to protein, gene expression, the central dogma of molecular biology. When a gene is switched on, an enzyme attaches to the start of the DNA sequence. It moves along the DNA, opening the helix and at the same time making a chain of mRNA, messenger ribonucleic acid. The DNA code determines the type of base that are added to the growing mRNA chain. This process of forming mRNA from the code of DNA is called transcription. The mRNA is processed and then it moves out of the nucleus where ribosomes, which are called protein factories, attach to the mRNA. The ribosome reads the three-letter code comprising of three bases in the mRNA to attach amino acids to form a chain. This process is called translation. There are 20 different kinds of amino acids, which are the building blocks of proteins, just like ATCG are building blocks of DNA. Once the translation of the gene is complete, the amino acid chain folds to form the 2D or 3D protein structure. The proteins perform different functions and are ultimately responsible for a particular characteristic or trait, like in humans, the eye color, or in plants, the leaf size or the flower color, etc. Biochemistry to biotechnology. We have seen that in any living DNA, a chemical is the instruction manual which determines the different traits, characteristics of the living organism. Now, genetic engineering, a biotechnological process, can be used to manipulate this DNA code and hence change the final characteristic. The manipulation can be by way of addition or deletion of DNA of a gene, or even by way of addition or deletion of genes in a genome. The gene can be cut from one or more species, modified to be suitable for expression in another species, and then introduced into such another species. The combination of DNA from different sources produces a completely new man-made DNA called recombinant DNA. The recombinant DNA technology leads to genetic engineering having various applications in agriculture, research, pharmaceuticals, etc. Examples: Let us take an example from the pharmaceutical industry. Human insulin. Scientists identified the human gene that codes for insulin, which is a protein produced in the human body, a higher or complex life form. The insulin gene sequence was manipulated to create a recombinant DNA sequence to be inserted in a bacteria, which is a microorganism, a lower life form. The manipulation was to ensure expressing the human insulin protein using the bacterial cell machinery. Since bacteria multiply fast and a lot of insulin can be produced by the genetically modified or transgenic bacteria, human insulin is now widely available. Research on higher life forms, such as animals, for clinical testing can be expedited genetically engineering as well. The first transgenic animal was a mouse. The uses of recombinant DNA technology in agriculture sector are many, such as marker-assisted breeding or creating transgenic plants for developing disease resistance. Papaya ring spot virus almost wiped out papayas in Hawaii, but the expression of a ring spot protein by genetically modifying papayas gave papaya resistance to the virus. Nutrition enhancement. Rice was engineered to produce golden rice, that is, rice having vitamin A beta-carotene, so as to help preventing blindness due to vitamin A deficiency. Golden rice was created by transforming rice with 2 beta-carotene biosynthesis genes. PSY, phytoene synthase from daffodil, narcissus, pseudonarcissus. CRTL, carotene desaturase from the soil bacterium Erwinia uridivora. The PSY and CRTL genes were transferred into the rice genome and placed under the control of an endosperm specific promoter so that they are only expressed in the endosperm. Modifications can also be made to make the plant herbicide resistant or produce plants that can survive drought, saline conditions, or plants with increased root, shoot, or fruit size. Creating man-made DNA. The gene of the desired trait that one requires to be expressed is first painstakingly identified by the scientists through a screening process. This may be from the same species or some other species or the modification of an existing gene. The gene may be modified by adding a promoter that makes expression in target species suitable. The gene may also be modified to make the protein fold in the correct form or be transported to the site of action in a cell or outside, etc. Once the DNA sequence is finally decided, it is to be inserted in the genome of the target species. Transgenic plant. The various processes for correctly introducing a correct foreign gene of interest into a plant to create transgenic plant of desired requirement is most crucial in the genetic engineering process. One of the methods that may be easiest to explain would be the use of gene gun on a plant. Gene gun fires a desired DNA as coated gold particle at the target cells with the hope that some would end up at the right place. Once the DNA is into the cells are grown using tissue culture techniques to develop entire plants, which are then transferred to soil and screened and selected for trait of interest. Bt technology. Bt toxin, a protein, is a biopesticide that kills some insects that feed on it, produced by a bacteria called Bacillus thuringiensis. This Bt toxin can be sprayed directly into the field. The scientists tried to introduce this into a plant cell to make them resistant to insects. The bacterial Cry2AB gene that encodes the Bt toxin was identified and its DNA sequence manipulated, attaching other pieces of DNA from other species that are necessary for the Bt toxin to be prepared and present in the plant at the right place, at the right level, and at the right time. The CRY2AB gene was, for instance, attached to a specific promoter derived from cauliflower mosaic virus for making it suitable for expression in the plant cell. It was also desirable that the toxin in the cell be transported to the plastids in the plant cell for better results, and thus the gene for the protein that would perform this function, the plastic transit peptide from Arabidopsis thaliana plant, was also added to the gene sequence. The final DNA constructed is therefore an artificially created molecule produced in lab which can be added to a plant cell, and later transformed plants may be produced from the plant cells. The transformed plant produces the Bt toxin, and the insects which feed on the leaves of such plant die, making them insect resistant. The whole process is genetic engineering, a modern biotechnology process performed in the lab.